bio PhD student working in the EKIMS lab. I am studying neuroscience, and specifically the development of neurocircuitry in the brain. So, today I'm going to present to you sort of the first step in neural transmission. So, as you know, the brain is made up of neurons, right? And neurons talk to each other so that you can function. And the way that they talk is they send electrical signals down these things they have called axons, right? Well, So originally it wasn't really known how these signals were sent down axons, but Hodgkin and Huxley, and I think in the mid-20th century, won the Nobel Prize for showing how this is done. What they found was something called the action potential. So what they found was that there was this repeatable, consistent change in membrane potential, so membrane voltage, in duration, and in size. So, how would an axon or a cell change in voltage? Well, we have two major players, sodium and potassium. So ions carry a charge. So if you move ions through something, you create a potential, correct? So, the players now, sodium moves in to the membrane, and potassium will move out of the membrane. Just so you know, imagine sodium going into the cell and potassium coming out of the cell. Right? So this stage here, the action potential, this change in membrane voltage, we'll call resting. Right? So at resting, you sort of have it. More potassium's leaving and some sodium's coming in, but there's no real change in the membrane voltage. Right? So upon some sort of stimulation, like we could say if you're touching something, pressure, or a molecule is binding a receptor that's opening up and now allowing ions to come through, you'll have an increase. So towards positive membrane voltage change, right? This is called threshold. Here I arbitrarily put negative 40 millivolts because I think that's what it is for like the squid giant axon that Hodgkin and Huxley got the Nobel Prize for studying. So once that happens, more sodium will flow into the membrane, right? So you have some sodium coming in because now ions coming into the membrane because there's some sort of change at the pressure or binding. And you have an increase towards a positive potential because you have positive ions flowing in. Meanwhile, potassium is sort of staying the same inside, leaving the membrane. It's not going out too much, right? So at the peak of the action potential, you have sort of a closing off of these sodium channels, and now all of a sudden these potassium channels open. So now you have a whole bunch of positive ions leaving, which returns the membrane back to its resting. This is resting. Right. So now, this is sort of abstract. Let me just erase the players here and explain to you now how that can move down an axon. Although if any of you have any guesses, you can feel free to guess. So this is generally, some dendrites, generally how an axon is shaped. You have a cell body, maybe two dendrites which receive a signal, an axon which will send a signal, and then this will connect to another cell, or how you send the signal. So let's say that you get sufficient depolarization or threshold right here, something called the axon hillock. You'll fire this action potential, right? Now, remember, you need to have a threshold in order for this to fire. So you'll get sufficient depolarization here, because there's an action potential right here, which causes this blip. So it's increased membrane potential, which can then fire another one, and another one, and another one. Yeah, you get the idea. And then down to the synaptic terminal, where then, upon sufficient depolarization and some other players, will release neurotransmitters or some other sort of signal to the next neuron. Any questions? Please, I'd ask a question.
So you're talking about how sodium and potassium are like released and taken in the various
Yes.
channels. So where are those channels located, like on your picture?
Along the axon, there will be channels. So generally, you have like spaces where there's myelin or some sort of insulation to keep this traveling quickly. And then in between the myelin, you'll have these pockets of channels, primarily sodium channels, and then there'll be some potassium flanking that.
Any other questions? Any comments about the presentation?
Take a deep breath.
This is so much more nerve-wracking than TA.
I don't.
Yeah, freshman.
Little comment for like partly visual learners, it might be nice to have like a little cell with potassium and sodium going in and out like here,
Okay.
explaining the graph,
Yeah.
like travel areas and stuff. They're on the cell with the ions going in and out.
Seems like. Something like that.
Yeah, that'd be good.
Yeah, just something to like show
Okay.
where what you're talking about is taking place in
Yeah.
context.
I had to guess about like what the axes were on the graph. Like I
Oh,
think,
sorry,
like, when you first
milliseconds
saw
to
it-
millivolts.
Yeah, like when you first.
I
it was,
messed
like,
up. I was
helpful.
supposed to say duration when I said consistent. I was supposed to say consistent duration,
Mhm.
and then this would be time if it's duration.
Yeah.
And I kind of messed that up. So, yeah.
I think it's like nice whenever you like present a graph, like you know, like this is time and this is going to be voltage.
Okay.
Yeah, it's easy to follow. It's easy to follow.
I feel like your diagram is good and it's like comprehensive and everything, but I feel like you were like crutching on the board a little bit because I don't feel like you were looking out at everyone, and maybe that was just me because I was behind you. But I feel
I don't
like
think
you
I
were
was
like
either.
focusing kind of like on the diagram, which is awesome. But I feel like everyone else was kind of like, I don't know. That's just
Yeah.
my feeling.
I agree, like more eye contact would be nice, and something to keep in mind is like whenever you're talking as you're writing into the board, it's really difficult to hear. Like this is a small room, so I don't think it was really an issue. But in a larger room, like in a lab, no one's going to hear anything you're
So
saying.
don't talk while I write.
It's difficult because it's like a
Would it
slower
have been okay for
pace.
me to come up here and draw this before I even started speaking? Because that's what I normally do.
That's fine.
And I was concerned because no one did that.
Yeah, I mean, obviously
like-
it's, difficult in this context,
Yeah.
and you can't come before class.
Yeah.
But just something to keep in mind now.